Next, we're going to be discussing the excitation of skeletal muscle, cardiac muscle, and smooth muscle. The process of stimulating muscle will cause contraction, and it is called excitation-contraction coupling. This process converts an electrical stimulus to a mechanical response. The stimulus is an action potential, and the mechanical response will be the contraction. So we will compare the three different muscle types and how they are excited. In skeletal muscle, excitation coupling relies on direct coupling between key proteins: the sarcoplasmic reticulum, calcium release channel, and DHPRs, which are a type of receptor. The DHPRs are located on the sarcolemma, while the sarcoplasmic reticulum receptors reside across the sarcoplasmic membrane. The close apposition of the transverse tubule and the two sarcoplasmic regions containing the receptors is described as a triad and is predominantly where the excitation coupling takes place. Next, we will move on to cardiac muscle. Well, first we'll talk about the source of calcium. For skeletal muscle is only the sarcoplasmic reticulum. And the effect that the nervous system will have on skeletal muscle is only excitation. So for cardiac muscle, unlike skeletal muscle, the excitation coupling is thought to depend primarily on a mechanism called calcium-induced calcium release. Though the proteins involved are similar, the DHPR and the sarcoplasmic reticulum receptors are not physically coupled. Instead, the sarcoplasmic reticulum receptors are activated by a calcium trigger, which is brought about by the activation of the DHPRs. Further, cardiac muscles tend to exhibit dyad structures rather than triad. So we can put that here, triad structure. The source of calcium for cardiac muscle is the sarcoplasmic reticulum and the extracellular fluid. And the effect that the nervous system will play on cardiac muscle is excitation, and it can also be inhibited. So let's move on to our last muscle, smooth muscle. So now we're going to talk about smooth muscle. It is important to note that the contraction of smooth muscle need not require neural input. That is, it can function without the action potential. It does so by integrating a huge number of other stimuli, such as hormones, metabolically using ions such as carbon dioxide and oxygen, or physical stimuli can also stimulate it, such as stretch receptors or shear stress. And when these are occurring, so the integrated, which include ions, hormones, physical stress, the excitation coupling will not be occurring here. Smooth muscle can, though, be excited by neurons. All neural input will be autonomic or involuntary. In these mechanisms of the excitation-contraction coupling, it will be as follows. The parasympathetic input uses the neurotransmitter acetylcholine. The acetylcholine receptors on the smooth muscle are of the muscarinic receptor type. As such, they are metabotropic or G-protein coupled receptors. The sympathetic input uses a different neurotransmitter, norepinephrine. So, parasympathetic uses acetylcholine, and sympathetic uses norepinephrine. All adrenergic receptors are also metabotropic. The exact effects on the smooth muscle depend on the specific characteristics of the receptor activated. Both parasympathetic input and sympathetic input can be either excitatory or inhibitory. The main mechanism for actual coupling involves varying the calcium sensitivity of specific cellular machinery. So the calcium source for smooth muscle is just like cardiac muscle. It's from the sarcoplasmic reticulum and extracellular fluid. And the effect that the nervous system will have on the smooth muscle will be excitatory or inhibitory. All right, the last topic that we will be talking about today is action potential types. So there are three main types: electrochemical action potentials, electrical action potentials, and intrinsic action potentials. For electrochemical action potentials, several neurotransmitters are used, which are derived from a class of molecules which we all know to be known as amino acids. The most common initiator of action potentials is electrochemical activity. This is caused when a cell is depolarized by a chemical signal. In neurons, this happens at special junctions between nerve cells, which we know as synapses. In the synapse model, a transmitting neuron releases a chemical signal called the neurotransmitter, which can be acetylcholine, norepinephrine, etc., which activates the receiving neuron by opening the special neurotransmitter gated ion channels, causing depolarization. If enough depolarization occurs, the change in voltage causes a population of special voltage gated ion channels to open near the base of the neuron. This is what initiates the action potential. Thus, an initial depolarization is triggered by neurotransmitters opening the ion channels, which in turn triggers an action potential via voltage-gated ion channels. This form of action potential is ideal for neurons because they can be readily strengthened or weakened, which is the basis for learning in our brains. The second type of action potential is electrical potential. Direct electrical stimulation can cause action potentials in neurons that normally will only respond to chemical stimuli. While electrodes can certainly be used to induce action potentials in brain cells, the nervous system actually already has its own type of electrode. Although most neurons are driven by chemical synaptic activity, some are activated by gap junctions. These are large pores that join two cells together. At these junctions, the electrical potentials move seamlessly from one cell to the other due to electrical current traveling uninterrupted within the cell's shared fluid. If this current induces enough depolarization in the downstream cell, an action potential continues to be done unabated. Some cells may require concurrent excitation by multiple gap junctions to activate. Our last type of action potential type is intrinsic action potentials. Ganglion cells of the retina encode varying degrees of color or brightness by firing at varying rates. Certain cells, such as bursting neurons of the hippocampus, do not require external activation to express action potentials. These cells have a unique phenotype that causes them to fire in periodic spikes or bursts. Rather than causing an action potential per se, the electrical chemical activity of presynaptic cells causes the intrinsic activity of the neuron to speed up or slow down. This type of signaling allows neurons to transmit complex signals encoded by the rate or pattern of firing rather than an all or nothing firing of a single action potential.